Okay, thank you for the introduction, Jeremy. It's wonderful to be back in Boston after a year's break last year, and we found a nice new venue for this conference, which hopefully we'll be continuing to use in some years to come. So in this talk, I'll go through some of the experience that we've had, I guess, working with a range of customers on their immune oncology programs, and it's an area that we've been involved with for quite some time, much before it was as exciting through the checkpoint inhibitors as it is now. So I'm not going to go through a long introduction of cancer immunotherapy. The idea, of course, is to exploit the specificity and memory of the immune response to fight cancer where existing approaches fall short. And we've all heard about the recent advances with checkpoint inhibition, which has really rekindled this field, although it was a proposition for many decades already that curing cancer through relevant immune responses might be eventually a good option. And perhaps it's also something that has already been working for us, you know, as a sort of outcome of, let's say, radiotherapy, perhaps even chemotherapy, where you induce cancer killing, and some patients do much better on those therapies than others. It's perhaps unclear whether those therapies really eliminate all of the tumor in each case. So, you know, how are those people who have a complete response respond? Perhaps there was always an immune response that has followed up in those populations, but that wasn't really recognized. So the checkpoint inhibition has really brought that back to the fore. But some major challenges remain. Long-term survival and quality of life certainly in that category, and also the effectiveness of the checkpoint inhibition is usually restricted to a smaller population. And the question is, well, how can that perhaps be expanded with additional therapeutic approaches that are more antigen specific and perhaps personalized, or at least, you know, falling into certain categories whereby cancers can be categorized and then be treated more specifically. So all of those perhaps improved approaches or potential approaches to cancer immunotherapy require a deeper knowledge of the actual molecular and cellular mechanisms of the immune response, which are not so well understood yet, although we have a sort of outline that there must be cytotoxic T cells, maybe helper T cells, perhaps also antibody responses involved. But really, there isn't a good understanding of exactly how that works and how it sort of segments out into individual antigens in each case. So in order to improve on that, we need to find more tumor-associated antigens, or whether they are self-antigens or mutated antigens, as explained in the presentation just now, and to be able to monitor the immune responses to those. So I won't go into this slide how checkpoint inhibition works. It's been talked about and explained very well in the talks yesterday and also today. There was a very good presentation by Nik Schwabe about this subject in 2014 at our conference, and Nik, who's a professor at Harvard, I believe was instrumental in developing the entire field. So that's on our website, and there's a very good review on all the ins and outs of checkpoint inhibition, and I recommend that talk for watching in some spare time perhaps. Jeremy has talked about the antigen presentation assay yesterday, which is really a workhorse assay for us, which we've introduced at this conference series in 2011. So we were really the first company to put out a commercial antigen presentation assay at that point in time. And we, I believe, are still leaders in that field and have now really numerous publications against it. There is a list on our website. The standard configuration that we've developed is more for the immunogenicity risk assessment, loading antigens onto cultured dendritic cells, where you can then recover presented antigens which are sequenced, and Jeremy has talked about that. But I will describe to you perhaps a bit more the opportunities in immune oncology and give you some examples of work that we've done with customers in this arena. So the thing to point out about ProPresent is really its versatility and flexibility. You can do just about anything you can imagine. So it's actually important to let your mind roam free a little bit on what is possible with this assay. Tumor tissue can clearly be used, like we've just heard in this presentation. When you have a tumor sample available for which you can produce an exome sequence, then basically that allows you to augment the search database for an antigen presentation assay, which normally would be the human genome. So you can add your mutated versions of the human genome to that and search the mass spec data from the tumor sample against those sequences. You can also use tumor cell lines. Sometimes tumor tissue from specific modalities or patients are hard to come by, and a good surrogate can be potentially a tumor cell line, and I will show that in a case study. You can also work with healthy donors, where you may then test the therapeutic approach, for example, immunization with viruses, and culture those, look at the antigen presentation that does. That was also covered in the Gritstone talk. And of course, once you have clinical trial or treated patient samples, they can also be analyzed. You can subculture cells, whether it's dendritic cells, B cells. The cell type is certainly not limiting for class 1, which is presented on all nucleated cells. And for class 2, you just have to make sure that it's a professional APC like a dendritic cell or B cell, or potentially a HLA class 2 transduced cell. In terms of the type of therapy or vehicle that you might be looking at as an antigen source, anything can potentially work that produces this antigen and gets it onto an MHC molecule, whether it's a sort of DNA or RNA type therapy. We've also worked with exosomes, viral vectors, even bacterial vectors, tumor lysates, and any form of protein, be they antibodies, ADCs, or more complex constructs, which are engineered in a variety of ways. And you then have the option to look at just plain vanilla peptides or mutated peptides, of course, but also modified peptides. So, for example, in cancer immunotherapy, potentially phosphorylated peptides that are differentially phosphorylated in tumor tissues could be relevant. There was a nice talk about that at our Mastering Immunity Europe conference at the end of March by Agnes, who are focusing on the tumor epitope discovery using mass spec approaches, and particularly looking at opportunities with phosphorylated peptides. Chemical modifications are also possible, and glycopeptides are perhaps more challenging, but in principle also a possibility, and something we will look more into in the future. The great thing about the methodology is that it's very separable. It's fundamentally divisible into 2 steps. One is to generate your cells, and the second step is to do the analysis. And the key thing is they don't have to happen in the same place. So we can do cell culture in a variety of modified ways to suit the program in our labs, and then follow on with the mass spec discovery. But equally, the customer can do that on their location with their samples in their context, just pellet and freeze the cells, and then they can be transferred in a dry shipper where they will retain all their function and MHC molecules. We then simply do the recovery at our site, and the peptide recovery from that, and the mass spec sequencing. So I'll give you some example of how that can work out in practice. So this is a case study for which we worked on with a small German company in collaboration with the German Cancer Research Institute as well. And this is an oncolytic virotherapy where we've used the ProPresent assay to find, first of all, some peptides that can be monitored, and then we also carried out the monitoring in the clinical trial. So this particular application was in glioblastoma multiforme, which is the most common primary brain tumor. And although it's not that common in humans, it is a very nasty disease with a very poor prognosis. So 50% of patients die within 12 to 16 months, and it has a very poor long-term survival outlook. So certainly significant clinical need. And the idea was to use oncolytic rodent protoparvoviruses as a cancer therapy. They are non-pathogenic for humans, and they're also very small viruses, so perhaps a little bit easier to handle from that perspective. Maybe less vector responses there potentially as well. They're highly oncoselective, cause direct oncolysis and were thought to have immune-mediated effects as well. So the therapy was to be administered as a combined intratumoral, intracerebral route and investigated, or to be investigated at the time, in a phase 1/2A trial with this PAV-ORIX-01 agent. So our job was to find some way to monitor the immune response and, first of all, figure out what is it that we are supposed to be monitoring here. So I guess it's quite typical of a program of that nature, which really has multiple constraints in terms of how you can set up a potential monitoring strategy. So first of all, we didn't have any patient cells available, of course, ahead of the clinical trial for epitope discovery. And even in the trial, the cell sample numbers would be quite low, so it's quite limiting. And to really discover some antigens that could be relevant, we therefore used parvovirus-infected glioblastoma cell lines, which were available to the customer, and that perhaps also drove some of the hypotheses behind the treatment strategy. And they were matched to the patient HLA that would eventually enroll into this trial. We then used the ProPresent assay to do a pan-class 1 MHC immunoprecipitation to find some new processed and presented peptides and resolve those peptide sequences, and found overall 5 peptides from 2 proteins, and there are only 2 proteins in this virus for the virus itself, and for the glioma condition, 20 peptides from 18 proteins. So we also used Elispot as an assay for this, and Elispot is a nice assay because it has such a low background and it can resolve the often low immune responses to cancer very nicely. And it's quite flexible in terms of how you use antigens to stimulate it. Of course, you can bias it more towards detecting either CD4 or CD8 cells. And what's really important to get it right is to make sure that all your cell processing is really set up correctly from the clinical center, then followed by a good cryopreservation protocol, and there's a properly qualified and validated assay, all of which we've gone through in our assay setup at ProImmune. So the specific monitoring that was then set up with the peptides that we found was to just do a plain vanilla gamma Elispot in a standard type setup using 4 pools of peptides, and those pools contained the glioma peptide, which were either found by ProPresent or which were in fact just identified from the literature as pre-existing known peptides from that condition that might be relevant, and the parvovirus peptides split into the protein source that they came from. In this case, we stimulated the cell culture slightly just to enhance the response a bit, and at a standard peptide concentration with typical controls like CF to look for a positive that's peptide specific, PHA as a sort of categorical positive, and media as a negative. And then significant responses are the mean minus 2 times the standard error of the mean should still be greater than twice the background. How exactly you set the background is a little bit up to you, but this is a commonly accepted way of doing it. So the monitoring and treatment setup was that at day 0, there was an intratumoral injection of the actual virus therapy, followed by a resection of the tumor and further intracranial administration of this vaccine at day 10, and then further monitoring time points at day 18 and day 61. And as you can see on the results, basically the controls behave as expected, so there's really no response with the negative and then beautiful positive responses as usual. And you don't see much of a response against these peptide pools through most of the monitoring time frame, but then good responses at day 61, certainly to one of the key parvovirus pools, which is very nice to see, but also relevant responses in the sort of 20 to 30 per million cell counts, which we still know are relevant in cancer immunotherapy and are responses are often seen at that level and can be important too. So that was a very nice outcome for this study, because we've basically gone from knowing really nothing about what we should be looking at in terms of the immune monitoring to finding relevant epitopes, synthesizing them, planning a strategy that was actually workable in this quite limited clinical trial, and then coming up with some relevant correlate data that this therapy was actually generating relevant immune responses. So the parvovirus isn't the only virus we've worked with. So this is a case study that isn't exactly in oncology, but nevertheless in looking at epitopes from viral vectors, and adeno-associated virus is also relevant to immune oncology. In this case, we worked with the Children's Hospital of Philadelphia, and here we use ProPresent to identify new AAV-derived peptides, and also potentially whether there would be any peptides from the factor 9, which was supposed to be delivered to hepatocytes. We didn't actually find any in this case for factor 9. But this was a scenario where the customer actually had given us the frozen cell pellets, and we then did the recovery and sequencing using ProPresent, and found significant peptides, which are probably A2 binding from the capsid in each case, and also other less significant peptides from the study. So those peptides were then confirmed by the group at CHOP using Elispot assay, and that's in a 2015 publication. So I'll jump a little bit over to the Pentamer technology, which has also been used extensively in, I guess, immune oncology, and we've been selling cancer pentamers to people since 2003, and people have worked on peptide vaccination for a long, long time and monitored immune responses to those kind of scenarios. But clearly there has been a renaissance in the arena since the immune oncology field took a leap a few years ago. So now more than ever, these reagents are highly relevant. So just to take you through the structure of the Pentamer again, it's a recombinant multimeric MHC molecule. In this case, it's pentameric, where we rebuild basically the MHC peptide complex that you would find on the surface of a cell with the antigen of interest. And the reason for having a multimer is so you get enough avidity to the TCR interaction, because it's a multivalent interaction naturally, and the affinity between, even for a good affinity peptide between the MHC peptide and the TCR is usually too low to stain it similar to a, let's say, antibody with just a monomeric form. So we are using a coil protein to basically malterize this molecule and give you the nice high valency. And it also gives us the opportunity to label the molecule through a labeling domain at the opposite end of the business end of this molecule, where we target fluorescent molecules through an antibody tag. So this technology has then been used in a case study that I'll take you through. This was a study in adoptive transfer for melanoma, malignant melanoma specifically, carried out by Marcus Butler, who was then at the Dana-Farber Cancer Institute. And it was a very nice study in terms of all the different follow-ups he did. So it's a great paper that is cited on the next page. So the idea was to do autologous adoptive transfer of patient-derived CD8s specific to the tumor antigen, and it's the very well-known MART1 antigen, which I guess is a self-antigen rather than a mutated antigen, but one where you can show that, I guess you can break the tolerance and have some relevant effective immune responses. And the major limitation, though, with these adoptive therapies where T cells are somehow augmented ex vivo, is that the response is still very short-lived, and so the objective was to increase the longevity of these T cells once they are transferred back into the patient. So the objective is overall to try and get an effector memory response that's long-lived and has the right effector memory phenotype, and that was achieved in part through peptide pulsing artificial antigen-presenting cells, which I believe had MHC monomers on the surface that would then bind a specific peptide, and they could, this was a bead-based vehicle which could interact with the autologous T cells and basically drive their proliferation in an antigen-specific manner, and with co-cultured with IL-15 and IL-2 to just make them last a bit longer. 9 end-stage melanoma patients were treated and infused twice at 35-day intervals with no further manipulation of host immunity at that stage. So this was before checkpoint blockade. And then the MART1 response was assessed with this Pentamer, which is, I guess, one of our favorite items, I would say. And you can see that just in one case, subject 7, there's a small pre-existing immune response, which you can see quite clearly with the Pentamer, but perhaps not sufficient, certainly, for that person to have resolved the melanoma. So these were mostly end-stage patients. And once you do the adoptive transfer, then, of course, you do see a very much enhanced population of antigen-specific T cells that you can easily monitor with Pentamers. And again, when you infuse some more cells at day 35, you know, that population-wise it seems larger, but percentage-wise it's about the same, slightly smaller actually. Unfortunately, though, as is commonly the case with these sorts of therapies, at day 70 and day 126, so relatively soon, you get just a big reduction of the T cell frequency. So it's still not tremendously long-lived. But when you then combine it with checkpoint inhibition in another case, you can see that at day 74, we're back to the same scenario where you started out perhaps with a, you know, smallish response. You've probably gone through infusion and reduction, so you're back down to that level. But if you then add back the anti-CTLA-4, then you can reboost that immune response. And I guess that's perhaps the beauty of the Pentamer technology, or Multimers in general, that you can follow very specifically what the CTLA-4 antibody does in this case, and how the response is enhanced back. And at a very late time point, it's still highly persistent and also highly clonal in terms of the V-beta chain usage of the TCR. And the follow-up also included that you can see the right sort of migration to the effector memory phenotype that was desired in this case. And as far as I know, there were some complete responses in that study as well, which of course is very hard to link that one-to-one to the response to that specific epitope, but nevertheless quite promising and correlating with the observed immune responses. A slightly different case study, just looking at peptide vaccination, but here the objective was to really evaluate the formulation of the vaccine. And I believe there's some attendees from Immunovaccine here with us as well. So we've been working with them for some time. This is a publication from 2012 by Berenstein et al. So it's just a straightforward 23 HLA-A2 restricted cancer patients of a variety of cancers. They received this Depovax formulation, which contains 7 A2 restricted peptides as antigens from a variety of, I guess, well-known cancer antigens. And the patients received the vaccinations 3 times over 3 weeks. And then again, immune monitoring was done with Pentamers and also ICS and Elispot. So this shows you some of the results that were observed. First of all, at different time points with a negative Pentamer, and then at day 0, 21, 42, and 73. You can see different patients listed here, and different Pentamers for different antigens shown here for different types of cancer. And so a twofold increase in Pentamer positive CD8 cells in this study was considered as a sort of positive response that could be generated in a patient. And 2 out of 3 patients had increased tumor antigen-specific immunity as a consequence of this vaccination. And so I guess that fulfilled the objective of demonstrating not just the usefulness of peptide vaccination with those antigens, but also the formulation in this case. I think what you do see with peptide vaccination, again, is this sort of augmentation and then slight decrease in many cases. But some people certainly had persistent responses that went to the end of the monitoring window as well. So I guess, an interesting study from that point of view too. About a year ago, or 2 years ago now, we've introduced class 2 tetramers, and prior to that we are still also offering class 2 monomers. We heard about the importance of having class 2 epitopes involved in generating a relevant immune response as well. And although in the immuno-oncology field the focus is very much on the CD8s and trying to get those cytotoxic cell-based responses going, we think that maybe, the difference in some of those patients that might get a complete response to some checkpoint blockade, maybe down to the fact that they also have a more complete immune response, perhaps through cytotoxic T cells, but hopefully also involving some helper T cells, and equally a humoral immune response, maybe where, again, helper T cells may be relevant in really sort of driving that response too. That's certainly the picture you would get from a vaccine scenario. And let's try and not call Andrew. Who knows? Anyway, so the class 2 products can shed more light on that in the future, we hope. We offer them for many different diseases, including many infectious diseases, but of course also for many different cancer antigens now. And our range of HLAs, which is at the moment DRs, is constantly expanding. So recently we've brought in some more sort of Asian-focused alleles. In oncology, it's interesting that the cancer burden is a little bit different out in Asia, obviously a massive market of the future, where things like EBV-linked cancers are, well, they're important here, but they're more important out in China and Southeast Asia for sure. So we can cover research into a different range of modalities. And just some data to show you that they do work well. They may need peptide stimulation to really get the frequencies up where you can resolve antigen responses at those levels. But yeah, we're basically here to sort of broaden that field and hopefully bring the interest and demonstrate the relevance of class 2 responses in oncology in the future. And I guess the monomers are also potentially of interest because people are using them in part to look at developing anti-MHC peptide-specific antibodies and perhaps use those to target cancer in the future. I wanted to touch on the fact that we can also change some of our key assays, like the DC T cell assay, which is set up very much as a sort of standard assay for looking at therapeutic protein immunogenicity. A very simple variant of that is to set it up as a mixed lymphocyte reaction. So the assay setup is very similar to the standard assay in that we still generate monocyte-derived dendritic cells in culture from a donor cohort, and then you just mix them with T cells or PBMCs from a different donor, which generates a massive cross-reactive response. And by mixing different donors off against each other, you can then get a good sort of cohort-broadened study going. And well, what's the point of it? Well, you can modulate it through some kind of immunotherapy, be it checkpoint blockade or some other immune modulator that's to enhance or reduce immune responses to specific antigens, and the effect can then easily be monitored looking at the differences in proliferation that's achieved over a 7-day window. In addition to that, we can add other biomarkers and other markers because it's set up as a flow cytometric assay, which is running on at least 8-color machines. So phenotyping of responses is also possible. We've had the question about regulatory T cells that can clearly be investigated, or whether it's a naive or a memory response that you're looking at. That's also easily worked out through phenotyping that you can carry out using flow cytometry. I just wanted to maybe talk a little bit about important points on the workflow that's undertaken when we design immune monitoring studies, such as a Elispot study, which for immuno-oncology, which we're getting into more and more. And as I've touched on, there are many challenges typically in those studies, mainly around the sampling method, the exact antigens that are to be monitored. And usually challenges with, not having enough cells, not having enough antigens to monitor, and working out, well, what can you do and how can you do it to get meaningful data, like, for example, in the Oryx case study that can work to the constraints of your particular project. So, of course, you need to decide which peptides to use or antigens that may be developed or derived from your therapy of interest, how exactly they are going to be split into pools and aliquoted to be used in the study, and those aliquoteds have to last you over the course of the study and not go off in the study period. We need to understand exactly where the positive controls perhaps are coming from. The CEF response may not be a good positive control in severely immune compromised patients, which may be the case in a cancer protocol. And then I guess you go through the specific setup and objective and patient source and think about how you're going to qualify the assay, starting from the cryopreservation protocol to the repeatability and robustness of control responses, and then exactly how will samples be taken, processed, then eventually shipped and received, and how are they going to be batched to then do the analysis either in one go or in several blocks. So typically we tend to end up with a sort of block-style analysis before we finally produce a final report and report on the study. And I think what I would say is the core part is to get this bit right, where you really try to understand, well, where are these cells coming from? What are you going to expect in terms of cell numbers? What's the, variability envelope of the cells that you are to expect from your study? It may be that you're targeting 10, 20 million cells per patient, but maybe what you will actually get ranges from 2 million or nothing to 40 million. So how are you going to prioritize maybe testing those? What sort of qualification study should you set up to make sure that your protocol works no matter what in practice is thrown at us as sort of outcomes from the collections, which can be pre-planned but don't always work entirely to plan. But one can sort of pre-plan those eventualities as well. And exactly how will they be shipped? What are the potential complications in shipping, receiving, time of shipment, receipt, customs clearance, etc. So it can be challenging, but providing you really think it through, identify all the risks, and sort of build the study design and qualification of your testing correctly, then that really increases your chance of success many-fold. And I would say that's an arena that we have quite a lot of experience in by now. So just to summarize then, better outcomes in immune oncology really require you to have a better understanding of how it all works, what mechanisms form the basis of an effective immune response in cancer. And we think that really requires much more detailed characterization of antigen-specific immune responses and finding the epitopes that are relevant. And of course, you need access to a wide range of specialist assays that really can give you the right understanding at the right time. And economics are important, so having a large toolkit available then allows you to pick and choose the best variants for that, for your particular objective. And so we have, wherever we can, try to enhance the tools ourselves, be it the Pentamers or Tetramers or the ProPresent assay and many other assays, to make sure that we really deliver you the sort of cutting edge in this arena. And by now, as Jeremy has also mentioned in the context of immunogenicity, but also cancer immunotherapy, I would say that we have encountered most common approaches that are out there. So each one of them has got their unique challenges, but at least having encountered them before, we have already experienced some of those challenges and had to overcome them. So the experience there is also key to success. So with that, I thank you for your attention and answer any questions that you may have.